Good morning, everyone. Guys, good morning. Guys, good morning. I am here already. Guys, good morning, everyone. Oh, good morning, my first listener. Okay, today we're supposed to discuss hepatitis viruses. Good morning, everyone. Start collecting, please. And we start with the hepatitis viruses. And a lecture before the last lecture, we studied picornaviridae family members and hepatitis A and E viruses. Why we divide those hepatitis viruses into two large groups? So, according to their ecological features or way of transmission. So that it means that hepatitis A and E are transmitted through the are called fecal oral hepatitis viruses. So to cause infection, they need to be ingested. So they cause infection through GIT. And that's because they are both icosahedral naked viruses with a tightly packed capsid, and they are highly resistant to the stomach acidity and intestinal tract enzymes. So they are not inactivated in the intestinal tract by enzymes. While the rest of the hepatitis viruses, B, C, D, G, at least these four, are so-called parenteral hepatitis viruses. What does it mean, parenteral? They are transmitted by any route except for GIT, because they are all enveloped viruses and they are highly sensitive to the organic solvents, acidity, enzymes that may inactivate the viruses. So parenteral means they may be transmitted through the sexual intercourse. They can be inoculated with the contaminated needle. They can be spread through the organ transplantation or blood transfusion. But all routes, but not through GIT, because they will be inactivated in GIT. So today we'll speak about those hepatitis viruses that are considered to be parenteral. And you can see here in the title slide the target organ of all those viruses, that is the liver. So they have very narrow tissue tropism, all those viruses, and that's what is common for them. They multiply in liver cells, in hepatocytes only. So, and usually not multiplying in other cells. So those hepatitis viruses have some common features, as we said. They cause an inflammatory response in liver associated with icteric symptoms. I hope you know already that icteric symptoms is the development of so-called jaundice, and you can see the liver from the case of hepatitis infection. Hepatitis viruses are readily spread because infected individuals are contagious before or without showing symptoms. That makes them so common in human pathology. But then in other features, all those hepatitis viruses are different. They belong to different families, and they have different structures, and they replicate in their own ways. And that's why they may cause specific symptoms development, and the time course and the severity of the disease can be different. So then from the point of view of immunity, they all are distinct, and they are different antigens by which we may recognize them. So when the serology of the disease, the course are different for every single virus. So let's start with just this table. You can see here the common table for both oral hepatitis viruses and parenteral hepatitis viruses, just with mentioning when they have been discovered. You can see that the first discovered was hepatitis B virus and hepatitis A virus. Then hepatitis E virus had been, hepatitis D virus had been discovered in 1977. Then HEV in 1980. And then we've got the hepatitis C virus, hepatitis TTV, and the hepatitis G virus, and the last one, hepatitis F virus. So that's still speculated what kind of virus is this, so we won't discuss it so much. And hepatitis A and B were the first hepatitis viruses appeared, and traditionally the rest of the hepatitis viruses, like C, D, E, G, are well known as non-A, non-B hepatitis viruses. So you can find this abbreviation somewhere in the book still. So as I said, fecal-oral transmission is typical for HAV and HEV, while the hepatitis that we discussed today are transmitted by other routes, and they are called parenteral hepatitis viruses because of the parenteral transmission. Here we see the comparing of the most common out of these four, because still hepatitis G is very rare. So the most common are B, C, D hepatitis viruses. And out of those, hepatitis B is double-stranded DNA virus, and hepatitis C, D, and also G are RNA viruses that have single-stranded RNA. So hepatitis D is also called delta agent because it is not complete virus, it is sort of defective virus that we discuss later on. And you can see here they all belong to different families. So hepatitis B is from Hepatnaviridae family from the genus Orthohepatnavirus. Hepatitis C from family Flaviviridae and genus Hepacivirus or Heparnavirus. That's some difference. And hepatitis D virus is a viroid-like structure. We'll discuss it a bit later on. Out of these three are all envelope hepatitis viruses with different set of surface glycoprotein that will be discussed in particular place when we discuss the particular hepatitis viruses. So they are all of small size. Hepatitis B 42 nanometers, hepatitis C is a bit larger, up to 60, and hepatitis D is also small, 35 nanometers in diameter. So they are all acid ether sensitive. So they can be inactivated quite quickly, but unless they are covered with the blood, my dears, as a part of the blood clot, they may not be inactivated very easy. So the prevalence of these hepatitis viruses is very high. The highest is for hepatitis B virus, a bit less for C, and prevalence of hepatitis D is regional. In some regions, it's very high. So what kind of infection they cause? All are transmitted through the parental route and sexual route. But the sexual route is not really very common for hepatitis C. It is believed that is the not really commonly transmitted by the sexual route, and most commonly through blood or organ transplantation. And take a look at the duration of the incubation period. It is very long, from several days, 14 days or 15 days for CAV, for up to several months. And usually, hepatitis B cause onset insidious, and so do the hepatitis C virus, but hepatitis D virus cause the onset that is abrupt usually. So if we characterize the diseases caused by this hepatitis virus with a short description. So hepatitis B infection is usually mild, occasionally severe. Infection with hepatitis C is usually subclinical or very mild, but then the consequences can be also very severe. And the infection caused by hepatitis B virus is usually very severe, with a high to very high rate of the death. The lethality is very low for hepatitis C and really very low for hepatitis V. So, and the ability to cause fulminant infection is very low in B and C, but very high in hepatitis D. And the time course of the disease is also different in these hepatitis viruses. So hepatitis B usually causes acute disease with the chronization of the process in 5 to 10% of the cases. Out of those 5 to 10%, in 10% of those, the cirrhosis will be produced. And in 80% of those who produce cirrhosis, it produces cancer, but then it is cancerogenic or oncogenic. So it is believed that hepatitis B is true oncogenic virus. If we take a look at hepatitis C virus, because it usually causes subclinical mild infection and the body won't produce vigorous immune response against hepatitis C. So then usually the disease is very mild or subclinical, but then 80% of cases develop chronic process. Out of those, 20% develop cirrhosis, out of which in 5% the cancer will be developed. But then the way how the cancer is developed is different from that of hepatitis B virus, because hepatitis C is RNA virus, so the RNA genome cannot enter the human DNA chromosome. So then it is believed that the cancer associated with hepatitis C virus infection is rather the result of the permanent inflammatory response in the liver. And the hepatitis D virus usually causes cirrhosis, fulminant, lethal hepatitis virus infection. So the important thing is that the very good vaccine is developed against hepatitis B virus, that is a genetically engineered subunit vaccine that is made up of recombinant hepatitis B surface protein or antigen. And the vaccine also against hepatitis B also protect human being from hepatitis D virus. Why? Because hepatitis D virus is a delta agent, cannot cause infection alone or solo. It needs help from hepatitis B virus, so it may cause only super or co-infections. I hope you remember these terms that we studied in general microbiology. So then, the vaccine is not developed against hepatitis C virus yet, and it is not available. But I would like to remind you from the very beginning that the good treatment is developed against hepatitis C virus infection, but not against hepatitis B virus infection. Few words about the significance of this group of viruses for the inhuman pathology. So if we calculate, so it will appear that around 500 million people are persistently infected with hepatitis B or hepatitis C, and are at risk of developing chronic hepatitis, cirrhosis, and liver cancer. And why it is so? Partially because there is the absence of cell culture system for both hepatitis B and hepatitis C viruses. And that is important because the modeling of the infection in animals help us to understand the virus life cycle and the basis for pathogenesis, and it also is required the culturing of the virus for development of the vaccines and antiviral drugs. So within last decade, a lot of research groups are working on the hepatitis viruses, and so they succeed in developing a novel human hepatoma cell line termed like this that have been established that supports the entire life cycle of both hepatitis B and hepatitis C. Why it is important to make a model of the whole life cycle? Because as a result, we may get the infectious virion, so the development of the virus from the very beginning to the very end when infectious virus particles are developed. By that moment, it was not possible. This cell line provides a very good tool for addressing the virus life cycle and the development of antiviral drugs and vaccines. But this culture or these cell types are not really used in the routine diagnosis of hepatitis virus infections, my dears. This is the only research tool by now. So then it means that culture method is not really applicable regarding hepatitis B, C, and D infections. So now we start our description with hepatitis B virus that usually infects liver, kidney, and pancreas on humans and chimpanzees. It is a small enveloped virus, and you can see that is the true virus that is circular, the spherical virus with this 42 nanometers diameter, and it is made up of partially double-stranded DNA, and it is a very small one, around 3.2 kilobases. And this virus encodes a polymerase with reverse transcriptase activity. It is a very important distinguishing feature of hepatitis B virus. And based on the sequence comparing, hepatitis B viruses are classified into 8 genotypes from A to H, and each genotype has a distinct geographical distribution. If you take a look at this particle, you can see that it is made up of core, made up of hepatitis B core antigen. This is a yellow here, right? And inside partially double-stranded hepatitis B viral DNA genome with the polymerase with reverse transcriptase activity. And this nucleocapsid is covered with the lipid bilayer with inserted there hepatitis B surface antigens that are shown here in green. So this is quite simple. But then if you take a look at the blood of infected individuals, you will see there not the only one type of particles where we can find the genome and the core and the polymerase. So this is infectious virus particle that is called Dane particle. That is of 42, 47 nanometers in diameter. So 7 because we may consider the length of the glycoprotein spikes. So, but then apart from this Dane particle, some other particles can be found. They are filamentous, you can see like this, right? And they are spherical, you can see like this, but without electrons dense core of the virus. So means they are empty particles. They have diameter 22 nanometers, and these spheres or filaments are composed of hepatitis B surface antigen only that is inserted into the host cell derived lipid membranes. So, and there's no nucleic acids inside, so it means those particles, these two, are non-infectious. And the only true virion that is called Dane particle is infectious because it contains genome, polymerase with reverse transcriptase activity, core, and surface glycoprotein, so all elements that are needed to perform replication in the host cell. And you can see here that these are the subvirional or subviral forms of hepatitis B, and this is particle usually outnumber the actual virions. You can see here very few true virions with the electrons dense core, and they are outnumbered with the not infectious virus particles. But those particles are highly immunogenic, so they may act as decoy for antibodies produced against hepatitis B surface antigen. So, in fact, instead of binding with Dane particle, so antibodies may bind with these spherical or filamentous hepatitis B surface antigen particles. So, and then they will be wasted for nothing because those are not infectious. So initially those particles had been used as the first vaccines against hepatitis B. But since we learn how to do it in the laboratory, we don't use them anymore. The infectious hepatitis B virion has spherical structure, 42 nanometers in diameter, and the envelope surrounds the icosahedral capsid made up of hepatitis C core antigen. Genome is partially double-stranded, and P or polymerase is covalently bound to the 5' end. And you can see that except for the hepatitis B, C antigen, we have very small amount of hepatitis E, HBE antigen. Because why so few molecules? Because the processing of hepatitis E antigen is a bit different. So it is produced inside the infected cell, and usually it is primarily secreted into the serum and can be found in the blood serum, and only very few copies can be found inside the infectious virion, while the hepatitis C antigen is not secreted to outside the infected cell, because this is a major capsid protein and is used to build up the nucleocapsid of the newly synthesized virions. So then, if we are talking about glycoproteins, we may distinguish LMS glycoproteins. Do you remember how you measure the size of the T-shots? SML. That's how we measure the size of the hepatitis B surface glycoproteins. All these glycoproteins are encoded by the same gene and read in the same frame. And this is a small glycoprotein, is contained in M glycoprotein, and M glycoprotein is larger, is contained in L glycoprotein. So we will see it right now. See? So in the Dane particle, we can have M, S, and L glycoproteins. And the large glycoprotein is an essential component for virion assembly. And these glycoproteins contain the group-specific and type-specific determinants. So the combination of these group-specific and type-specific determinants result in development of the 8 subtypes of hepatitis B virus, as we said already, that those hepatitis B viruses have different distribution within the world. So here you can see all proteins that are found in the patients that are infected with hepatitis C. The only one, hepatitis B X protein, is missing. So this is a not secreting protein, but this protein is minor protein, and it is believed that it is responsible for the oncogenic transformation of the cells that harbor hepatitis B virus. So now we discuss the way of hepatitis B virus replication, because, my dears, this is rather sophisticated, and this is a unique way of replication with a lot of distinguishing features. So first some most common distinctive characteristics. Hepatitis B has a defined tropism for liver. Why it is so? Because now the specific receptor is found, hepatitis V, that is called sodium, and this is an sodium taurocolate co-transporting polypeptide. So it is used as a cellular receptor. And then another very important property, that hepatitis B, do you remember, this is DNA virus, but it replicates through the RNA intermediate. That is a unique also. So then hepatitis B virus produces hepatitis B surface antigen containing particles. As I said, they act as antigenic decoy for protective antibodies. And the hepatitis B virus X protein is essential for virus infection and has been implicated in the development of the liver cancer. So, a liver-specific bile acid transporter named the sodium taurocholate co-transporting polypeptide has been identified as the cellular receptor for hepatitis B and its satellite, the hepatitis D virus. It likely acts as a major determinant for the liver tropism at the entry level. So it means the virus may enter those cells, they keep those receptors. You can see this is the receptor. And it is believed that the heparin sulfatation inhibitor receptor can be used to prevent the binding and entering into the cell. The viral envelope usually fuses with the cell membrane. So we describe the replication of the virus, right? So you can see here that the virion binds with NTCP receptor. And we believe that it fuses with the cell membrane, releasing the nucleocapsid into the cytoplasm. And the virus then will be decapsidated, as you see here, and genomic genomic double-stranded DNA will be released from the virion and transferred into the nucleus. Then, what event is the very important one? So first, what should be done? So repair of the double-stranded DNA. It should be built up into completely double-stranded DNA molecule. And then this double-stranded DNA molecule will be subsequently supercoiled to form so-called CCC DNA, covalently closed circular proviral DNA. You can see it here. See? Covalently closed CCC DNA, right? And then usually this CCC DNA is formed under which hepatitis B virus persists in the host cell. And in this form, in the form of supercoiled, covalently bound circular DNA molecules, it is not reachable for all factors of immunity. So, in this form, it may persist for a very long while. But then, because it is not integrated into the cellular genome, it can be transmitted to progeny cells. It serves as a reservoir for viral replication when antiviral therapy is interrupted. The supercoiled DNA then can be transcribed by cellular RNA polymerase into mRNA and into mRNA. Then this mRNA will be taken out of the nucleus into the cytoplasm, and then translation of these transcripts will occur on the cellular ribosomes. Then the supercoiled DNA molecules also can be transcribed into pre-genomic RNAs. What is it? Pre-genomic RNAs. We mentioned that the hepatitis B virus replicate through the RNA intermediate. So pre-genomic RNA will be produced. So then they will be encapsulated into the cytoplasm together with the molecule of hepatitis B and DNA polymerase. The latter has a reverse, so the DNA polymerase has reverse transcriptase function that catalyzes the synthesis of the negatively stranded genomic RNA. That's what we can see here. So then, when the RNA, pre-genomic RNA, together with polymerase, are packed into the core, the only there the DNA synthesis will occur inside the packed already core where the nucleic acids are packed already. As soon as negative strand DNA is produced using our pre-genomic RNA as a template, then immediately the pre-genomic RNA will be digested, because as far as we know that there should be no DNA and RNA together in the viruses, right? So the RNA will be completely denatured. And then what happens? So positive DNA strand will be synthesized by the polymerase using negative strand DNA as a template. But newly generated nucleocapsids can be recycled to yield additional CCC DNA molecules in the nucleus, but most of them bud into the endoplasmic reticulum to form mature virions that are subsequently released into the pericellular space by exocytosis. So from this we see, my dears, that by budding through the endoplasmic reticulum membranes, the viruses obtain the surface glycoproteins, and then they are released into the environment by exocytosis. But by the way, can you see here that overproduction of surface antigen makes possible the forming of this intermediate circular or filamentous hepatitis B surface antigen particles. Can you see that also separately from the DNA particles, these particles, subviral particles, will be released by exocytosis through the cell membrane. So we can see as a result the development of the completely done DNA particles, then subviral particles. So when the DNA particles will start to be released, the production of the second DNA positive sense molecule will be stopped immediately. So that's why usually it will not be time enough to build up the complete positive sense DNA strand. So that's why when viruses are released, the positive strand is usually incomplete. So we've got partially double-stranded DNA molecule. That is the short description of the viral hepatitis B replication. If we take a look at the possible targets that can be used in treatment of hepatitis B infection, you can see here that possibly the inhibitors can be used that are mircludex B and cyclosporin A. There are the sodium taurocolate co-transporting polypeptide inhibitors. They may inhibit this receptor and prevent so the attachment of the hepatitis B virus to the surface receptor. Then the nucleotide analogs can be used to stop the reverse transcription, and that is also one of the possible ways to stop the viral replication in the infected cell. So, but apart from this, we don't see any much targets for treatment of the hepatitis B infection. So that's why until now we are not very good in treatment of hepatitis B. So within the circulation of these covalently bound DNA molecules, the event that is integration of the hepatitis B genome into the host cell DNA may happen, and it is usually associated with long-term infection, and then it may cause hepatocellular carcinoma development. The transformed cells usually express hepatitis B viral antigens. Rearrangement and juxtaposition of the viral promoters next to cellular growth controlling genes occur, and the viral X protein may act as transactivator of transcription. It turns on the transcription of the not only viral proteins but also cellular proteins and promote and stimulate the cell growth, so that may lead to the oncogenic transformation of those cells. Few words about the pathogenesis of hepatitis B virus infection. You can see here that the transmission may occur through a lot of routes, with mother's milk, vaginal secretion, blood, semen, saliva, because everywhere in all those fluids, the hepatitis virus particles, DNA particles, can be found. As soon as the virus enters the bloodstream, so it circulates within the bloodstream and enters the liver through the NTCP receptors. And it establishes the infection in liver, and the surface antigen will be released. Quite often, when the antigen is released, it is highly immunogenic, so a lot of antibodies will be produced. And hepatitis B surface antigen, so-called Australian antigen, and its antibodies, because the overproduction of the antigen and antibody have tendency to form so-called immune complexes that can cause very severe complications, and that is so-called immunopathological reactions as a result of the immune complexes circulation in the bloodstream. So then the release of the DNA particles causes viremia, and because of this viremia, virus can be found again in any secretion in the body that may cause infection, including neonates infection. And also it is very common in blood, in those who need blood transfusion or organ transplantation, or intravenous drug users, so where the virus may be injected directly into the bloodstream. So then here you can see that the presence of antibodies are very important, because as soon as the virus enters the bloodstream, but any of the routes, so if antibodies are present in the bloodstream, they can prevent spreading of the virus into the liver, so they may prevent the spreading or the development of the disease. And also the presence of antibodies or cell-mediated immunity may help in resolution of the disease before the newly synthesized DNA particles will be released into the bloodstream. So usually source of infection is any fluid of infected individuals. Root of infection is the inoculation, sexual contact, or through the child birth. So the character of disease can be acute or chronic, symptomatic or asymptomatic. And mainly the character of the disease depends on the dose of the antigen or dose of the virus, and it depends very much on the person's immune response to the infection that we discuss right now. So, and the symptoms may not be observed for 45 days or longer, but then at the end of the incubation period, the person can start to be infectious. So the infection proceeds for a long time without causing liver damage or symptoms. That's because hepatitis B virus is not cytopathic virus. So it doesn't cause really very pronounced CP effect. But then the copies of the hepatitis B genome integrated into the cell chromosome may remain for a very long while. Cell-mediated immunity and inflammation caused the symptoms and the resolve of the infection. So what does it mean? If the immunity is very strong, developed against hepatitis B virus, so most probably we will have the liver damage, but then we have also the resolve of the infection. And of course, it takes a very long while for parenchyma to regenerate, but then it will be complete recovery. And the insufficient T cell response, cell-mediated response, usually result in the occurrence of the very mild symptoms and the development of chronic hepatitis. You can see it here. So if the infection is multiple and effective cell-mediated immune response is developed, so it causes liver damage and the damage to the virus-infected cells, and the symptoms will be quite severe with jaundice and release of liver enzymes into the bloodstream, but most probably we have resolution of the infection. When the dose of the antigen or dose of the virus was lower or something wrong with the cell-mediated immunity, it is limited formed against hepatitis B virus, then the hepatitis B virus will be able to persist in the liver cells. See, some of them will be killed, but not all. This persistence of hepatitis B virus causes so-called chronic disease with very mild symptoms, maybe without jaundice. And then it may occur for a very long while. If we have the co-infection or superinfection of this carrier with the hepatitis D virus, so-called delta agent, most commonly it may cause fulminant hepatitis disease. So, but then even the persistence of hepatitis B virus in the liver may cause cirrhosis and primary hepatocellular carcinoma. That is the most common. Antibodies generated by vaccination are very protective against the infection. They can block the infection because they will be neutralizing antibody. They will prevent the entry of the virus, the binding of the virus with the anti-CP receptor, so it prevents the infection. But then the bad thing about about the anti-HBs antigen antibodies is that the immune complexes can be formed, and they may form hypersensitivity reactions of type 2 and 3 that may result in vasculitis, arthralgia, rash, and renal damage. Usually, infants and younger children exhibit less tissue damage and milder symptoms because they don't have very effective immunity. And that's we discussed already. Two major causes of chronic infection with hepatitis B are the persistency of the covalently closed DNA molecules in the nucleus of hepatocytes, and the second is local immune tolerance to the hepatitis B infected liver. See the global prevalence of hepatitis B with the Russian Federation, the percent from 2 to 5, and even high in some parts of Africa and Asia. So, and it is found almost everywhere. Who are at risk, my dears? If you take a look at medical students, they are of very high risk. Some of them can be drug users or multiple sex partners, and they are medical students that may be exposed or at work. So then it is very important to be vaccinated. So because the vaccine is developed. So how do we perform laboratory diagnosis of hepatitis virus? My dears, first, of course, the clinical symptoms. But it's good and liver enzymes, so-called biochemical blood tests, right? But then it is not enough for differential diagnosis, because these are just symptoms of hepatitis that it may be caused by some other factors, like toxic hepatitis, right? Or hepatitis C virus may also cause sometimes the same symptoms development. So that's why for definitive diagnosis, we use serological tests, and we use ELISA tests usually. And we're supposed to find within these ELISA tests so-called serological markers. My dears, what are serological markers of hepatitis B virus infection? These are the hepatitis B virus proteins that are antigens and antibodies against these proteins, both types of serological markers. The antigen themselves and antibodies against these antigens can be used as serological markers, but not all hepatitis B antigens have same diagnostic value. For example, hepatitis B E antigen is very important because it is secreted antigen, and it will be usually secreted in large amount into the bloodstream during the virus replication. And the finding of hepatitis B E antigen best correlate with the presence of infectious virus particles in the blood and not just persistent infection, while the finding of hepatitis B surface antigen may be during the persistency also. So antibodies against hepatitis B antigen are of high diagnostic value, especially the antibodies against hepatitis B core antigen. Do remember that core antigen itself is not secreted to the environment, to the blood. So that's why the hepatitis B core antigen is not used as serological marker, while the antibodies against this antigen are of very high diagnostic value, especially if we find IgM antibodies. That will be a clear sign of acute current hepatitis B virus infection. You can see here the most typical dynamic of the markers of hepatitis B infection within the acute hepatitis and within the chronic hepatitis B. Of course, the DNA of hepatitis B in both acute and chronic hepatitis can be found but by polymerase chain reaction. But then it will be almost indistinguishable. So to identify the type and stage of the disease, we rather use the serological markers. So the thing is that during the symptomatic phase of an infection in acute hepatitis, we may have the so-called window period when antibodies are not produced yet. And also, when antibodies start to be produced, we may not find HBs antigen because the antigen-antibody reaction may cause the development of the immune complexes. So we will have a gap in identification of HBs antigen and anti-HBs antibodies. Within the chronic infection, it is usually characterized by the continued finding of hepatitis E antigen, hepatitis S antigen, and both, and the lack of detectable antibodies to this antigen. See, anti-HBs, so no anti-HBs. How about treatment of hepatitis B infection? The thing is that if there is a sort of algorithm how to manage it. So usually if we have acute hepatitis B infection, we won't perform the antiviral treatment because we believe that the body is fighting with the hepatitis B virus itself. And the treatment is usually performed in some cases of chronic hepatitis virus infection. For example, if DNA is lower and there are no liver enzymes released into the bloodstream, so usually treatment is not performed. Then when the viral load is very high in the blood and the very high level of IL-10, IS-10 liver enzyme, then the treatment supposed to be performed. So then, if the level of hepatitis B viral DNA is lower and the liver enzymes are normal, the liver biopsy should be performed. If no or mild histological disease, so no treatment is usually performed. If moderate or severe histologic changes in the liver occurs, then the treatment is supposed to be performed. Now, my dears, few words about hepatitis C. So, that's for treatment we use. What drugs are approved for hepatitis B? Interferons. Why interferon can be used? My dears, do remember that interferons are effective against RNA viruses, but not DNA viruses. But it is partially effective in combined antiviral immunomodulatory therapy with the immune interferon, because these viruses replicate through the RNA intermediate. So that's why pegylated interferon alpha can be used for treatment now, or conventional interferon alpha with direct antiviral activity. Then the second group of drugs are nucleoside or nucleotide analogs. And these are the drugs produced that have direct antiviral effect, but unfortunately the hepatitis virus may produce resistance very quickly against these nucleoside analogs. So the prevention is done by general prophylaxis that is seen here, but most probably by the vaccination, that is specific prevention. Vaccination in all countries worldwide are recommended for all infants, very small babies. This is one of the very first injection or vaccination within the baby's life. Then for older people, it is recommended for people in high-risk groups known to be anti-HBs, antigen negative. Vaccination is useful after exposure for newborns or hepatitis B surface antigen positive mothers and people exposed to blood or secretion from infected persons. Immunization of mothers is also recommended. So this is subunit genetically engineered vaccine because it what kind of genetic engineering is performed? Plasmid containing S gene for hepatitis B surface antigen is cloned into the yeast Saccharomyces cerevisiae, and then when Saccharomyces are cultured, they secrete a so-called HB virus surface Australian antigen. It is purified, isolated, purified, and used as an antigen in vaccination. Now, my dears, hepatitis C virus, family Flaviviridae, and genus is Hepacvirus. It is spherical with 60 nanometers in diameter, and the envelope is made up of the cell membrane and the envelope glycoprotein that are called E1 and E2 that are usually form dimers on the surface of the envelope. And inside we have single-stranded DNA molecule. And antigen or serologically, the hepatitis C virus may be classified into C genotypes. They are distinguished by serology. And around 170 to 200 million individuals have hepatitis C virus. That represents around 3% of the world population, including 3 to 5 million people in the United States. According to the WHO Regional Office in Middle East, Egypt has the highest prevalence in the world with 7% of prevalence in adults. You can see it here, right? But then you can see here the distribution of different type of genotypes is not also same. So the most common is genotype 1. In Russian Federation, in the United States and North America and South America also, but not in Africa, where the genotype 4 is the most common. Then genotype 3 is also found almost everywhere except for particular places in Africa. Now structure. This is the very simple structure of the hepatitis C virus. And you can see here the viral genome. And it starts with the internal ribosome entry site that is used as the uh, priming site of the transcription. And around 7 proteins, non-structural proteins, are encoded by the genome, and 4 structural proteins that are nucleocapsid, envelope glycoproteins E1 and E2, then ion channel protein P7. So then we have all those NS. These are non-structural proteins. The proteolytic processing of the polyprotein. Why we show here polyprotein? Because the translation occurs from the whole genome into the polyprotein. And the cellular peptidases and 2 viral proteases produce the processing of the polyprotein and break it down into active proteins. Subunit C, E1, and E2 form the virus particles. C, they stand for course. P7 may have a function in assembly. It is not shown, it is found here. So, then non-structural proteins from NS3 to NS5B form the viral replicase complex and important in viral genome replicases. NS5B usually forms the RNA-dependent RNA polymerase that is responsible for viral genome replication. Few words about the replication of the viral genome. It appears to be very sophisticated and very complicated. So hepatitis C virus exists as a... So then, one more thing I would like to mention. We mentioned that the virus causes very weak immune response. One of the reasons why, so because in a body it exists as a lipoprotein virus hybrid lipoviral particle, that is LP, or lipoviral particle. It is covered with low density phospholipids of the human body. And then it doesn't cause the vigorous immune response, that's why. And the scavenger receptor class B type, that is the scavenger receptor of B1 type, is the molecule that is important in lipid trafficking and tetraspanin CD81. They used as a receptors by hepatitis C to enter the host cell. So it means the hepatitis virus uses the receptors responsible for trafficking of the lipids in the cell, and then they enter by receptor-mediated endocytosis into usually clathrin-coated pits, and due to the acidification with P7 protein of the endosomal environment, the fusion happens and the viral genome will be released into the cell. So you can see here that the nucleocapsid with the viral genome then later on is released into the cell. Oh, sorry. Sorry, just a minute. That, alô? Okay, you can see here that as soon as positive sense genomic RNA is released, so polyprotein translation occurs because RNA is recognized directly as the mRNA. And then the viral proteins are produced. And then the viral assembly occurs with the positive sense produce copies of the RNA molecules, and viral assembly occurs when the viruses travel in Golgi apparatus to get enough phospholipids into the glycoproteins and phospholipids into the envelope. And then the virus will be released by receptor by the exocytosis, the way that is very typical for viruses that don't cause CPE effect. So it causes the very mild damage to the host cell, so without pronounced CPE effect. Maybe that's also one of the reasons why they don't cause very vigorous immune response. And then new virions will be produced. The viral cycle is not that sophisticated. So, the characteristic of the infection. Usually, hepatitis C viruses spread by the same routes as HBV, but sexual contact transmission is very rare. So viremia can be detected within from 1 to 3 weeks of HCV transmission. And it may last for 4 to 6 months in people with an acute infection, or longer than 10 years when a persistent infection occurs. See, my dears. So within 10 years, the person may be infected, have persistent infection without developing specific symptoms that will be important for recognition of hepatitis C infection. So, sometimes hepatitis C occurs as acute disease, and the symptoms will be similar to acute hepatitis A or B infection. The inflammatory response, although is less typical and less severe to compare with the hepatitis B or A infection. It doesn't really cause fulminant fatal disease. Chronic hepatitis is the most common type of the infection. And the chronic persistent hepatitis usually or quite often progress to cirrhosis. Cirrhosis occurs in 20% of the chronic individuals, and the liver failure occurs in 20% of cirrhotic cases. And as we said, hepatitis C virus is oncogenic. How do we perform laboratory diagnosis of hepatitis C infection? My dears, ELISA serological tests are less important in diagnosis of the disease. First, seroconversion may occur very late in the disease. Do you know what is its seroconversion? When the person starts to develop antibodies. It may occur within 7 to 31 weeks of the infection. See? So the antibodies may delay. So that's why serological tests will be not really very important. But usually antibodies are present and quite often may not antibodies may not present in people where viremia can be seen. So that's why the most accurate, specific, and reliable test for hepatitis C infection is the polymerase chain reaction. That is the best indicator of the disease, and within which we can detect the RNA of the virus can be detected in those people who won't develop immune response against hepatitis C virus. So, and the thing is that is the general idea of the disease. So, in HCV infection, they only 30% are ecteric. Out of them, 80% out of the infected individuals, 80% cause chronic hepatitis. Out of them, after 10-30 years, from 30 to 50% develop cirrhosis. If we use interferon alpha, we may decrease the percent of those who produce cirrhosis. And in 2 to 6% of those who produce cirrhosis, the hepatocellular carcinoma can be developed. So interferon, of course, can help. But thank God now we have a quite good combination therapy of interferon alpha and ribavirin before, and now except for ribavirin or instead of ribavirin, we can use first or second generations of the antiviral drugs produced against hepatitis C virus. So these are boceprevir and telaprevir, both of these inhibit the non-structural protease. And then the second generation is sofosbuvir, is a nucleotide analog of the cellular uridine nucleotide that inhibits the polymerase that make RNA copies for newly synthesized viruses. And it is, we believe that the polymerase inhibitors can be very effective in combination with the pegylated interferon and ribavirin. So, simprevir also now is available, and this is the second generation of the hepatitis C antiviral drugs that can be used with a very high effectiveness from 95 to 100%. I don't know whether we reach this 100% or not yet. So, but then we believe that these drugs are very effective against hepatitis C. So, my dears, if we compare hepatitis B and C, for hepatitis B we have not so good treatment, but very good vaccination. And vice versa, for hepatitis C virus, we have no vaccination and no vaccine is developed yet, but quite good new drugs for treatment. So that is how we are going to control the hepatitis C infection, right? So you can see here the places where, or the targets that are used. So we may have non-structural 3 to 4 protease inhibitors, and you can see here, and we have non-structural 5A inhibitor. These are those that inhibit the polymerase activity, and is a polymerase replication activity here also. So, and we think that due to these drugs, we may control the hepatitis C infection. Now, a few words about hepatitis D virus. That is called the delta agent. It is RNA virus. The virion is spherical, and it is enveloped virus. The virus is acid sensitive and prevalence is low. And here you can see the structure of this virus. And it replicates only in people infected with hepatitis B infection because it is a parasite of hepatitis B, because it's been using S, M, and L HBs antigens from hepatitis B to make the envelope. So, my dears, these proteins are not encoded in the single-stranded RNA molecule, the genome of the virus. So, but then inside we have two types of hepatitis D antigen. Large hepatitis B antigen, this is the blue one, and navy blue small hepatitis D viral antigen. So, inside we have single stranded circular RNA molecule, very small one with 1,700 base pairs that usually fold into a rod-like structure due to self-complementary. And hepatitis D antigen thus forming the ribonucleoprotein here, surrounded by HBs antigen envelope protein. And this is the helper hepatitis B virus. That is served as a donor of M glycoprotein, S glycoprotein, and L glycoprotein. See? From here to here. Like this and like this. Then it makes the envelope. This is a characteristic of the hepatitis D virus infection. And this virus is cytotoxic. It means it has direct cytotoxicity upon the infected cells. So, and the combination of hepatitis D and hepatitis B virus infection cause usually very severe liver damage. Hepatitis D dramatically increases the severity of hepatitis B infection and usually cause fulminant hepatitis virus with the hepatic encephalopathy, massive hepatic necrosis, and in 80% of those co-infections, it is lethal. How do we perform lab diagnosis of hepatitis D? The delta antigen and antibodies against this delta antigen can be detected by ELISA or radioimmunoassays. And there is no specific treatment against hepatitis D virus infection. But then this infection, as I said, is preventable by the immunization against hepatitis B virus infection. Because as far as hepatitis B won't occur in the body, there will be no donor of the envelope glycoproteins for hepatitis B virus, so it cannot multiply there. Few words about hepatitis G virus. It belongs to family Flaviviridae, and we believe that the virion is spherical, 60 nanometers in diameter, and it has an envelope. The genome is single-stranded RNA molecule, and because of the envelope, the virus is ether and acid sensitive. The prevalence of this virus is moderate, and you can see here that it has the major capsid protein and the nucleic acid inside the icosahedral capsid. and the envelope glycoprotein. So, the hepatitis G virus is transmitted like other parenteral hepatitis viruses and can be identified by the only polymerase chain reaction, because serological tests are not really accurate if developed against it. The capability of hepatitis G virus to cause fulminant or fatal diseases is uncertain by now. So, and also vaccines are not available against hepatitis G virus. My dears, with this I would like to finish. So then, see you in classes. Goodbye, my dears.